Long ago in ancient Africa, a special family of early humans lived. They were called Australopithecines, and they are an important part of our history. Imagine a world very different from today, with wide-open grasslands, thick forests full of mysteries, and ancient lakes shining under a hot sun. From Lucy to Australopithecus sediba, we're going on a big journey. Their story shows us where we came from. We'll learn about the Australopithecines, our old relatives who came before us. These early human-like creatures were part ape and part human, and we can learn a lot from their fossils. We'll see how they lived, changed, and helped make way for humans. The term Australopithecine comes from an old way of grouping these ancient relatives. Scientists now divide them into two main types: the gracile Australopiths, Australopithecus species, and the robust Australopiths, Paranthropus species. In this video, we'll be focusing on the gracile Australopiths. We'll look at them in order, starting with the oldest Australopithecus species and moving to the youngest. But keep in mind, these species didn't simply appear and disappear one after another. Some lived for longer periods than others, and many existed at the same time in different places. We'll start with Australopithecus anamensis, the oldest known species of Australopithecus, living during the Pliocene era between 4.2 and 3.8 million years ago. Nearly 100 fossil specimens from over 20 individuals have been found in Kenya and Ethiopia. The first fossil, an arm bone, was discovered in 1965 by a Harvard research team led by Brian Patterson in Kanapoi and Alia Bay, northern Kenya. However, Patterson couldn't identify the species from this single bone. In 1994, a research team led by paleoanthropologist Meave Leakey found numerous teeth and bone fragments at the same site. Leakey and her team identified these as belonging to a primitive hominin and named the species Australopithecus anamensis. Though considered more primitive compared to other Australopiths, anamensis had knee, tibia, and elbow parts that suggested bipedalism. The tibia bone of anamensis had a more expansive upper end with bone, indicating its adaptation to walking on two legs. Despite this, evidence from the fossils also suggests that anamensis was capable of tree climbing. Evidence from fossils further show that anamensis had long forearms and modified wrist bones, indicating that it could use its upper limbs for support in trees or on the ground. The forearm bones measured between 265 mm and 277 mm in length. The curved proximal hand phalanx with strong ridges further suggests its climbing abilities. The cranium of Australopithecus anamensis combined ancestral features like a protruding face and a long, narrow brain case with a somewhat wide jaw joint that was flat from front to back, resembling the curvature found in great apes. Partial skulls suggest that anamensis had a relatively small brain of about 370 cubic cm, smaller than all later Australopiths and slightly less than the average chimpanzee today. Additionally, the ear canals of anamensis were narrow, similar to those of chimpanzees, unlike the wider ear canals of later Australopithecus and Homo species. The first lower premolar of anamensis had a singular large cusp, and its first milk molar was narrow with a large dominant cusp, possibly used for crushing. Fossil evidence indicates a high degree of sexual dimorphism, meaning males were significantly larger than females. This size difference suggests a competitive nature in mating and social dominance, similar to modern apes. Plant and animal fossils, along with the analysis of ancient sediments and rocks, provide insights into the environment of anamensis. They lived near an ancient inland lake in the basin where Lake Turkana is now, an area subject to volcanic activity. Volcanic eruptions produced layers of ash that covered the ground, and the fossilized remains of anamensis were found trapped between these layers, allowing reliable dating to between 3.8 and 4.2 million years ago. These early ancestors lived in forests and woodlands near the lake. Their skeletons show that they walked on two legs on the ground, but likely spent a lot of time climbing trees, possibly to find food or avoid predators. Their teeth indicate a diet of plant material, including fruits and hard-to-chew foods like nuts. While no evidence of culture has been found for Australopithecus anamensis, they may have used simple tools similar to those used by modern chimpanzees, such as twigs, sticks, and other plant materials for basic tasks, including obtaining food. Following Australopithecus anamensis in our evolutionary timeline is the famous Australopithecus afarensis, best known for the iconic fossil specimen Lucy. Lucy is a significant fossil find, consisting of several hundred pieces of bone, making up 40% of a female skeleton. Discovered in 1974 in Ethiopia by paleontologist Donald Johansen, her discovery provided crucial insights into human evolution. As one of the most complete ancient hominid fossils, Lucy offers a glimpse into early bipedalism, a critical step in human development. Australopithecus afarensis lived from about 3.9 to 2.9 million years ago in East Africa. They are among the longest-lived and best-known early human species, with paleoanthropologists having uncovered remains of more than 300 individuals. This species survived for over 900,000 years, which is about 4 times as long as our own species has been around. Afarensis had a tall face, delicate brow ridge, robust jaw, and prognathism, meaning the jaw jutted outward similar to gorillas. Their brains were about 1/3 the size of a modern human's, averaging around 430 cubic cm, similar to modern apes but larger than Australopithecus anamensis's brain. The brain's reorganization may have begun with some enlargement of parts of the cerebral cortex, indicating increased intelligence compared to their ancestors. A juvenile specimen's small hyoid bone, which helps anchor the tongue and voice box, suggests afarensis had a chimp-like voice box, likely unable to produce human-like speech. There was significant sexual dimorphism in body size, with males standing about 4 and 11 inches tall and weighing 100 pounds, and females standing about 3 and 5 inches tall and weighing around 62 pounds. Males typically had large crests on their skulls, which females lacked. The leg bones and some 3.6 million-year-old footprints made by afarensis in Latoli, Tanzania, indicate that afarensis was a competent biped, though not as efficient at walking as modern humans. The arm and shoulder bones shared similarities with those of orangutans and gorillas. This has been interpreted as evidence of either partial tree-dwelling ability or as basal traits inherited from the chimpanzee-human last common ancestor without specific adaptive functionality. Australopithecus afarensis grew up quickly after birth and matured earlier than modern humans, similar to chimpanzees. Because of this fast growth, they had a shorter childhood. This meant they may have had less time for parents to teach them and for learning social skills from others. Their quicker maturation likely affected how they developed socially and mentally, setting them apart from later human ancestors. Afarensis lived in a variety of habitats, including open grasslands, woodlands, shrublands, and forests near lakes or rivers. They were likely generalist omnivores, eating a mix of C3 forest plants such as figs, wild cherries, acorns, and flowers, and C4/cam savanna plants like grasses, seeds, roots, and succulents. They may have also eaten creatures like termites that fed on these plants. Carbon isotope analysis on teeth from Hadar and Dakika specimens, dated between 3.4 and 2.9 million years ago, suggests a diverse diet. Forest-dwelling individuals preferred C3 plants, while those in bush or grassland areas favored C4/cam plants. This indicates that afarensis could exploit various food resources across different habitats. For example, in times of scarcity, they may have relied on hard, brittle foods like nuts and seeds, as suggested by their dental anatomy. Fossil animal bones with cut marks found in Dakika in 2010 provide evidence that afarensis included significant amounts of meat in their diet. These cut marks suggest that they used simple tools to process animal carcasses, similar to the way modern chimpanzees use sticks to extract insects. This dietary flexibility would have been crucial for their survival in the varying environments they inhabited. Australopithecus afarensis had a large cone-shaped rib cage, suggesting a diet of high-volume, low-quality foods. Their skull structure indicates front teeth were primarily used for food processing. They likely used simple, unmodified tools from their environment, such as sticks and stones. Evidence from Dakika, Ethiopia, dated to 3.4 million years ago, shows cut marks on fossil bones, implying the use of stone tools for flesh removal and possibly marrow extraction. However, no modified stone tools have been found from this period. Australopithecus afarensis likely lived in small, mixed-sex social groups. The species exhibited significant sexual dimorphism, with females being considerably smaller than males, a trait common among Australopithecines. In 1995, scientists found a new type of ancient human relative in Chad, Africa. They named it Australopithecus bahrelghazali. These creatures lived about 3.5 million years ago. This discovery was exciting because it showed that these early human ancestors lived in more parts of Africa than we thought before. We don't know much about bahrelghazali because we only have a few jaw pieces and one tooth. Their teeth look a lot like those of other similar species, with big front teeth and wide back teeth. We can't say much about their brains, but they were probably about the same size as their relatives. One interesting thing about bahrelghazali is that their chin sticks out a bit more than other species like them. This makes them look a little more like later humans. These ancient relatives lived near lakes, surrounded by forests and grassy areas. By studying their teeth, scientists found out what they ate. They mostly ate foods from the grasslands, like grasses and roots. They also ate some forest foods like fruits or insects. This shows they could eat many different things, depending on what was available. We don't know much about how Australopithecus bahrelghazali lived day to day, but we can guess they probably acted like other similar ancient human relatives living in Africa at the same time. They likely used simple tools like sticks or other plant materials they found around them. They might have used stones as tools too, but we haven't found any proof that they shaped or changed these stones. We think they lived in small groups with both men and women, and both grown-ups and kids. This is similar to how many animals and early humans lived. Right now, we're just making educated guesses about a lot of this. We need to find a lot more fossils and other clues to really understand how bahrelghazali lived. Scientists hope that in the future, they'll discover more remains of these ancient relatives. This would help us learn more about them and fill in some of the blanks in our human family tree. Around the same time as Australopithecus afarensis and Australopithecus bahrelghazali, there's Australopithecus deyiremeda, an extinct species of Australopithecine found in Ethiopia's Afar region. This species lived about 3.5 to 3.3 million years ago during the Pliocene epoch and was identified in 2015 based on jaw fossils. Its holotype is an upper jaw with teeth, and there are two lower jaws and a small fragment of another upper jaw as paratypes. Because only 3 partial jaw bones have been found, it's uncertain if these fossils belong to a distinct species or are part of Australopithecus afarensis. This uncertainty has sparked debate among scientists. However, paleontologists argue that the differences in dental features, cheekbone position, and jaw robustness justify classifying deyiremeda as a separate species from afarensis. Deyiremeda is considered part of the diverse late Pliocene Australopiths. Deyiremeda is distinguished from Australopithecus afarensis by its thick enameled teeth, the more forward position of its cheekbones, and its robust lower jaw. Its smaller anterior teeth suggest a different diet, likely involving tough foods like sedges. This species' strong jaws and thick enamel indicate adaptation to a diet primarily consisting of such resilient plant materials typical for Australopiths. Living alongside Australopithecus afarensis, deyiremeda may have practiced niche partitioning, where competing species use different aspects of the environment to coexist. Despite sharing the same habitat, their dietary and habitat preferences may have differed, possibly due to genetic differences or adaptations to specific ecological niches. Similar to chimpanzees and gorillas, which share habitats and diets, deyiremeda and afarensis likely consumed similar foods when available and turned to different fallback foods during scarcity. While no evidence of tool use has been found, like other Australopithecines, they probably used or made simple stone tools. Next is Australopithecus africanus, which lived in South Africa between 3.3 and 2.1 million years ago. They were one of our early human relatives, first discovered in 1924 when a child's skull was found in a quarry. At first, scientists didn't believe it was related to humans because its brain was small, but later discoveries proved its importance. These creatures had a mix of ape-like and human-like features. Their brains were larger than chimps, but smaller than modern humans. They had big back teeth with thick enamel. Males were typically about 7 inches tall, and females about 4 tall. 1. Africanus could walk on two legs but wasn't as good at it as we are. They also had some features that made them good at climbing trees, like curved fingers and long arms. Their hands could grip things precisely, which might have helped them use simple tools. They lived in areas with both woodlands and grasslands. They ate a variety of foods, including fruits, leaves, and plants from the grasslands. They might have eaten some meat too, probably by scavenging rather than hunting. We're not sure exactly how they lived in groups, but some evidence suggests females might have moved between groups to find mates, similar to how chimpanzees do today. Africanus likely used simple tools like sticks or stones they found around them, but we don't have evidence that they shaped these tools. Next on the timeline is Australopithecus garhi. Australopithecus garhi lived about 2.5 million years ago in what's now Ethiopia. We don't know exactly how long they existed because we've only found a few fossils. These fossils are important because they help fill a gap in our knowledge of human evolution between 2 and 3 million years ago. Scientists first found part of a garhi skull in 1997. Since then, they found a few more bones. Garhi had a small brain, about the size of a chimpanzee's, and a jutting jaw. Their back teeth were quite large. We think females might have been around 4-7 inches tall, but we're not sure if males were larger. Garhi could walk on two legs like humans, but they could also climb trees well. Their arm bones were more like apes, but their leg bones were long like humans. This might mean they could take longer steps when walking. These creatures lived at a time when the environment was changing. Forests were becoming less common, and grasslands were spreading. Interestingly, some of the oldest stone tools we've found come from the same time and place as garhi fossils. We've also found animal bones with cut marks from stone tools near garhi remains. This suggests that garhi might have been one of the first of our relatives to make and use stone tools and to eat meat regularly. If garhi did indeed make and use these tools, it would be a big deal. Before this discovery, scientists thought only later human relatives in the genus Homo could make tools. The evidence suggests that garhi might have carried their tools with them to use multiple times. Australopithecus sediba, found in South Africa, marks one of the last parts of the Australopithecus story, existing towards the end of this genus's long history. Discovered in Malapa Cave, South Africa's cradle of humankind, Australopithecus sediba is represented by a juvenile skeleton, MH1, and an adult female skeleton, MH2. Fossils of this species date back between 1.95 to 1.78 million years ago, providing a glimpse into a specific time frame rather than its entire existence. Sediba lived alongside Paranthropus robustus and Homo erectus. The well-preserved skeletons from Malapa Cave offer scientists a detailed view of what individuals looked like around the time of Homo's evolution. Sediba exhibits traits similar to earlier Australopithecus species, such as tooth details, limb proportions, and a narrow upper chest. Conversely, its tooth characteristics and broader lower chest resemble those of humans, providing insights into the origins and connections within the Homo genus. Changes in Sediba's pelvis suggest an adaptation for upright walking, aligning with Australopith traits. Measurements of bone strength in the humerus and femur indicate a more human-like locomotion pattern compared to fossils attributed to Homo habilis. These findings indicate Sediba regularly walked upright, with pelvic changes occurring before other anatomical shifts seen in later Homo specimens. Australopithecus sediba was one of the last species of Australopithecus. It lived about 2 million years ago in South Africa. Scientists found really well-preserved fossils of sediba, which helps us understand a lot about them. Sediba had a mix of old and new features. Its skull looked a bit more human-like than earlier species, with smaller back teeth and a face that didn't stick out as much. Its brain was still small, like other Australopithecus species, but shaped more like a human brain. These creatures were about 4 to 5 feet tall. Their arms were long, like earlier species, but their hips showed signs of better walking on two legs. Interestingly, they had a unique way of walking. They turned their feet inward with each step. Sediba lived in an area with both grasslands and woods. They probably ate fruits, leaves, and maybe small animals. We don't have evidence that they used tools, but they might have used simple things like sticks or stones they found around them. Australopithecus species, including Sediba, are important because they show how our ancestors changed over time. They help us understand how we evolved from ape-like creatures to become human. These species lived between about 4 million and 2 million years ago, filling the gap between our earliest ancestors and the first humans. Scientists think Australopithecus evolved from an earlier group called Ardipithecus. These earlier ancestors could both climb trees and walk on the ground, showing a transition to a more human-like way of moving. Each discovery of Australopithecus fossils helps us piece together the story of human evolution. They show us the slow changes that led to the emergence of our own genus, Homo, and eventually to us, Homo sapiens.